All right, so let's talk about the humoral response. So again, we're talking about the adaptive immune system. This is the response that deals with or is mediated by B cells or B lymphocytes, and it's targeting extracellular pathogens. So these are pathogens that are, floating around in the body outside of cells. All right, so when we have our B cell, when the B cell is done with its education and it gets released into the body, it goes and hangs out in like lymph nodes and places like that, where it's more likely going to find its specific antigen that it's looking for. Remember that the receptors are antibodies. So they look maybe like that, and there's all over there here. And all of these antibodies have the same receptor. It's very specific, only looking for one type of antigen. So it looks kind of like that. And before it finds its antigen, it's considered a naive cell. So it's called a naive B cell. So this is a cell that has not come into contact, not found and bound its antigen. So B cells, basically, they hang out in lymph nodes or in the digestive tract, anywhere that it's a good place to find their antigen. They hang out there and look for it. And when they find it, they bind to it. So the first thing that happens is the B cell binds to its antigen. So let's say, here's a little, bacteria thing. So here's the pathogen. And pathogens have little markers on their cell membrane called antigens. And let's say this antigen is for here. So it's going to bind inside this receptor site on the antibody, which is the receptor of the B cell. So once it binds, it will endocytose this whole thing into the cell. So it will endocytose that. So number 2. So the B cell will endocytose the antigen with the antibody. So it brings the whole complex inside the cell. Okay, so then it goes through, it puts it in a vesicle, it sort of digests it, and it breaks down this whole thing into its protein parts. Then it goes through all of that, and then it posts that antigen on an MHC2 platform because this is a B cell, and a B cell is an antigen presenting cell. So it will do its thing, and then it will put its little MHC2 platform out there, and inside it is the pathogen. It's basically presenting that pathogen or antigen on its membrane. So now we have a B cell. The B cell has found its antigen. It's bound to its antigen. It's now presenting it on its plasma membrane. It's still not activated yet. Okay, it's still not really doing anything. So let's put this here. So it posts the antigen on MHC2, and it's presenting. This is an antigen presenting cell, and it is presenting this antigen to another cell. So then we still need activation for anything to happen. So we get another cell. Sorry. Let's do this. So we get another cell here, and this is a lymphocyte. And this is a T lymphocyte, and it is a helper cell. A helper T cell. And it has its T cell receptor is going to look something like that, so that it's going to lock onto this MHC with this antigen there. So it recognizes the MHC2, it sees its own antigen, and it's going to link there. Okay, so the helper T cell binds to the antigen on MHC2. Okay, so that happens, right? And then we get some little chemicals that come from the cell, the T cell, over to here. And these are cytokines that come from the helper T cell. Because it's bound, and yet we're still not activated yet. So these cytokines, these are called interleukins. And there's different numbers of interleukins, and I don't know what numbers these are, but it doesn't really make a difference. But these are interleukins chemicals. They're a type of cytokine, and they come over here to the B cell, and that's how it gets activated. And it activates the B cell. So once this thing is activated, let's come down here. So number 5. So activation, once it's activated, it goes through clonal expansion. And the B cell, what occurs during that is the B cell divides, and produces plasma cells. And the plasma cells make antibodies. So once it's activated, it will divide, and it will make these things. Let's put a different color. Well, let's make it this color. So it makes these little plasma cells. And these plasma cells then just barf out a bunch of antibodies. Along with that, so the antibodies, they go out, and the antibodies go out, and then they deal with the antigen, the pathogens that are there. Okay, because they're going to go bind to all the pathogen antigen receptors, and then deal with clearing them from the body. All right, so these are plasma cells. And it also makes, I'll do this a different color. It also makes memory cells. So these cells live on in the body, and when you're exposed again, then these cells immediately react. This process of activation takes time. Okay, so it takes weeks. The memory cells act immediately. Because they already know what to do. They've already been primed. Okay, they've already been through all of this. So they know what to look for and know exactly what to do. So that's the humoral response. B-cell mediated extracellular pathogens. All right, so next we'll talk about T cells and cell-mediated immunity.